Welcome everyone. Today, we delve into one of evolution's most extraordinary creations, the corpse flower. This isn't just any plant, it's a grand paradox, unlocking secrets about Rafflesia, the world's largest, stinkiest, and most mysterious bloom. Imagine a plant with no leaves, no roots, and no stem. For most of its life, it remains completely invisible, hidden from sight. And perhaps most famously, it smells of death to create life. Join me as we unravel the enigma of the Rafflesia. Let's meet this monster in the rainforest. The most iconic species, Rafflesia arnoldi, is truly massive. It can grow up to a meter in diameter and weigh over 11 kilograms. Its unique pollination mechanism, known as sapromyophily, relies on pure deception. It meticulously mimics the scent and appearance of rotting flesh to attract carrion flies, its primary pollinators. Crucially, there's no nectar reward here, it's a clever trick to ensure its survival. This remarkable plant is endemic to specific regions, as shown on the map, making it a rare and precious find. To understand Rafflesia, we need to consider its anatomy, or rather, its lack thereof. Unlike a normal autotrophic plant with leaves for photosynthesis, a stem for support, and roots for nutrient uptake, Rafflesia is an anatomical ghost. It has no chlorophyll, meaning it cannot photosynthesize. It also lacks any conventional plant structure, no true stems, no true roots. This makes it a holoparasite, meaning it is 100% dependent on its host for all its nutritional needs, living entirely within another plant. So, how does such a plant survive? It does so by living as a plant within a plant. Rafflesia exists primarily as an endophyte, a cryptic network of cellular strands that hides within its host. This infection phase involves uniceriate filaments stealthily growing within the host's vascular cambium. Its vegetative phase is entirely invisible, lasting anywhere from months to even years, without any external signs. Finally, when conditions are right, buds emerge, resembling cabbage-like swellings, eventually leading to the spectacular bloom that lasts a mere 4 to 8 days. For most of its existence, it remains hidden from our view. Rafflesia's unique lifestyle has led to what we call a genomic identity crisis. It is one of the only plants known to have completely abandoned its chloroplast genome. Even more fascinating, Rafflesia engages in extensive horizontal gene transfer, essentially stealing genetic blueprints directly from its host, the tetrastigma vine. This phenomenon highlights how evolutionary convergence acts as a bridge between the parasite and host, blurring the line between predator and victim in a remarkable genetic dance. Despite its incredible reproductive output producing seeds in the thousands, Rafflesia presents us with the great germination cold case. Its seeds are recalcitrant, meaning they cannot be stored for long periods. The profound mystery is that germination within the host has never been observed in the wild. And even with our best efforts, standard botanic garden protocols, using hormones and specialized soil, consistently fail to induce germination. We're missing a crucial piece of the puzzle on how this plant starts its life cycle. This brings us to decoding the transcriptome, where we gain critical insights. When we compare the genetic pathways of a model plant like Arabidopsis, a parasitic weed like Striga, and our enigma, Rafflesia, a key difference emerges. Striga, like many parasitic plants, relies on detecting strigolactones, chemical signals from host roots, to trigger germination. However, breakthrough research by Molina et al. In 2023 revealed that Rafflesia lacks the D14 gene, which is crucial for smelling these strigolactones. This means that synthetic hormones effective for other parasites are completely useless for Rafflesia, highlighting its distinct and mysterious germination requirements. This new understanding provides a new roadmap for resurrection. Our genomic studies show high expression of genes related to fatty acid and lipid metabolism. This leads to a compelling hypothesis: if not strigolactones, then what triggers germination? Our next steps focus on two primary suspects for chemical triggers: first, lipids and fatty acids themselves. Second, the presence of the KAI2 gene suggests Rafflesia might respond to characins, which are compounds found in smoke, or similar chemical signals. This opens exciting new avenues for research into germinating this elusive plant. Unfortunately, Rafflesia faces a severe threat, leading to what we call the silent erasure. A staggering 60% of the 42 known species are now critically endangered. Major threats include logging and palm oil plantations, which destroy its delicate rainforest habitat. Compounding this, its absolute host dependency means that if you destroy the tetrastigma vine host, you inevitably destroy the Rafflesia. This crisis often leads to invisible extinction. Taxa are being eradicated before science even has a chance to name and fully understand them. This slide highlights why ex situ conservation is failing for Rafflesia. For most plants, standard conservation in botanic gardens is relatively easy. However, the Rafflesia presents a unique challenge because it's a tripartite holobiont. We must simultaneously manage the parasite itself, its host vine, and its complex microbiome. While grafting has seen rare successes, like at the Bogor Botanic Gardens, it doesn't solve the fundamental seed puzzle. Moreover, there's a high mortality rate, with up to 73% of buds dying before they even get a chance to bloom. The Rafflesia challenge is truly hard. Amidst these challenges, indigenous stewardship offers vital solutions. Guardians of the forest, such as the Orang Asli and other indigenous groups, possess deep ecological knowledge passed down through generations. For them, Rafflesia are not just flowers, they are viewed as indicators of forest health and are often associated with forest spirits. This underscores a crucial conclusion: conservation efforts for Rafflesia, and indeed for many species, must be community-led. These local communities are truly the eyes and ears of the habitat, and their wisdom is indispensable. Ecotourism presents a complex paradox. On one hand, it offers a strong economic incentive, generating income for local communities and encouraging the protection of host vines on private land. On the other hand, it carries significant ecological risks. Uncontrolled foot traffic can trample delicate buds, and soil compaction can damage the ecosystem. The solution lies in initiatives like Pokedarwis or tourism awareness groups. These community-led regulations are transforming former poachers into protectors, ensuring that tourism benefits both people and the planet. The Rafflesia has been dubbed the panda of the plant world. We must leverage its iconography and charisma to drive funding for broader rainforest protection efforts. Our holy grail is to unlock the secrets of fatty acid germination, enabling us to grow Rafflesia from seed in captivity, a feat not yet achieved. Furthermore, we must designate its habitats as high conservation value HCV areas, ensuring these critical forests are protected and preserved for future generations. The Rafflesia blooms for only a few days, a fleeting moment of beauty, yet its survival requires a commitment for centuries. Saving the Rafflesia isn't just about preserving a single, magnificent flower, it is about saving the complex, often invisible web of life that sustains the entire tropical rainforest. Our call to action is clear: protect habitat, empower locals, fund science. These three pillars are essential for the survival of this grand paradox and the vibrant ecosystems it represents. For those interested in delving deeper, here are some key sources and further reading that informed today's presentation. These papers highlight the latest research on Rafflesia's genomics, its mysterious developmental stages, and the critical conservation challenges it faces. Thank you.